Genetic Control of Protein Synthesis, a Deep Dive. This presentation explores the intricate mechanisms by which genes govern protein synthesis. We will delve into transcription, translation, and the regulatory elements that ensure precise protein production. Understanding these processes is fundamental to comprehending cellular function and genetic diseases. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, mRNA. Here's a simplified overview. Steps of transcription. 1. Initiation. The process begins when an enzyme called RNA polymerase binds to the DNA molecule. 2. Unwinding. The DNA double helix is unwound, and one of the DNA strands serves as a template. 3. Synthesis. RNA polymerase reads the template DNA strand and matches the incoming nucleotides to the base pairing rules, AT and GC, with uracil replacing thymine in RNA. 4. Elongation. The mRNA strand is synthesized as the RNA polymerase moves along the DNA template. 5. Termination. When the transcription is complete, the mRNA strand is released. Key points. Transcription occurs in the nucleus, in eukaryotic cells. The resulting mRNA molecule is complementary to the DNA template strand. mRNA carries genetic information from DNA to the ribosome for protein synthesis. mRNA processing is a crucial step in eukaryotic gene expression. After transcription, the pre-mRNA undergoes several modifications to become mature mRNA, ready for translation. Here's an overview. Steps of mRNA processing. 1. Splicing, removing introns, non-coding regions, and joining exons, coding regions, together. 2. 5' capping, adding a modified guanine nucleotide, 5-cap, to protect the mRNA from degradation and aid in translation initiation. 3. Polyadenylation, adding a poly, A, tail to the 3' end, which helps stabilize the mRNA and facilitate translation. Importance of mRNA processing. Stability. Processing helps protect the mRNA from degradation. Transport. Mature mRNA is transported out of the nucleus and into the cytoplasm. Translation. Processing ensures the mRNA is ready for translation into protein. Key points. mRNA processing occurs in the nucleus. Proper processing is essential for efficient translation and protein production. Translation is the process by which the mRNA molecule is decoded to build a protein. Here's a simplified overview. Steps of translation. 1. Initiation. The mRNA molecule binds to a ribosome, and the first tRNA molecule carrying the amino acid methionine attaches. 2. Reading codons. The ribosome reads the mRNA sequence in codons, 3 nucleotides at a time. 3. tRNA selection. tRNA molecules carrying specific amino acids bind to the ribosome, matching their anticodons to the mRNA codons. 4. Peptide bond formation. Amino acids are linked together by peptide bonds, forming a polypeptide chain. 5. Elongation. The ribosome moves along the mRNA, adding amino acids to the growing chain. 6. Termination. When a stop codon is reached, the polypeptide chain is released. Key points. Translation occurs on ribosomes in the cytoplasm. tRNA molecules bring amino acids to the ribosome, matching codons to anticodons. The sequence of amino acids determines the protein's structure and function. Important concepts. Genetic code, the set of rules that dictates which amino acid is encoded by each codon. Reading frame, the sequence of codons is read in a specific frame to ensure the correct amino acid sequence. tRNA, transfer RNA, plays a crucial role in translation, acting as an adapter molecule that links the language of nucleotides to amino acids. Here's how tRNA works. Key functions of tRNA. 1. Amino acid attachment. Each tRNA molecule is charged with a specific amino acid by an enzyme called aminoacyl tRNA synthetase. 2. Codon recognition. tRNA molecules recognize specific codons on the mRNA through their anticodons, ensuring the correct amino acid is added to the growing polypeptide chain. 3. Delivery. tRNA molecules bring the attached amino acids to the ribosome, where they are incorporated into the polypeptide chain. Important features of tRNA. Specificity: each tRNA molecule is specific to a particular amino acid and codon. Anticodon: the region of tRNA that pairs with the mRNA codon. Amino acid attachment site: the site where the amino acid is attached to the tRNA. Role in translation. Accurate translation: tRNA ensures the correct amino acids are incorporated into the polypeptide chain according to the mRNA sequence. Bridge between nucleic acids and proteins: tRNA translates the genetic code into the language of amino acids. Regulation of protein synthesis is crucial for fine-tuning gene expression, allowing cells to respond to changing environments, developmental cues, and stress. Here's an overview. Levels of regulation. 1. Transcriptional regulation, controlling the rate of transcription, determining which genes are expressed. 2. Post-transcriptional regulation, regulating mRNA processing, transport, stability, and translation. 3. Translational regulation, controlling the rate of protein synthesis, often through regulatory sequences in the mRNA. 4. Post-translational regulation, modifying proteins after synthesis, affecting their activity, localization, or stability. Mechanisms of regulation. Transcription factors, proteins that bind to specific DNA sequences, enhancing or inhibiting transcription. MicroRNAs and siRNAs, small RNAs that regulate gene expression by binding to mRNA, often leading to degradation or translational inhibition. mRNA binding proteins, proteins that interact with mRNA, influencing its stability, localization, or translation. Phosphorylation and other modifications, post-translational modifications that can activate, inhibit, or alter protein function. Importance of regulation. Cellular adaptation allows cells to respond to environmental changes, stress, or developmental signals. Development and differentiation, regulated protein synthesis is crucial for the development and maintenance of cellular identity. Disease prevention, dysregulation of protein synthesis can contribute to diseases such as cancer, metabolic disorders, and neurodegenerative diseases. Key concepts. Feedback loops, regulatory mechanisms that adjust gene expression based on the levels of proteins or metabolites. Signal transduction pathways, cascades of molecular interactions that transmit signals from the environment or other cells to regulate gene expression. Protein folding is the process by which a protein structure assumes its functional shape or conformation. The role of quality control mechanisms in the cell is crucial for ensuring that proteins are correctly folded and functional. Importance of proper protein folding. 1. Functionality. Correct folding is essential for a protein's biological function. 2. Stability. Properly folded proteins are more stable and less prone to degradation. 3. Interactions. Correct folding enables specific interactions with other molecules. Protein folding process. 1. Translation. The polypeptide chain is synthesized. 2. Folding. The chain folds into its native conformation, often with the help of chaperone proteins. 3. Quality control. Cellular mechanisms check for correct folding and function. Quality control mechanisms. 1. Molecular chaperones, proteins that assist in the folding process and prevent aggregation. 2. Protein degradation pathways. Misfolded or damaged proteins are targeted for degradation by systems like the proteasome. 3. Endoplasmic reticulum (ER), quality control. The ER monitors protein folding and targets misfolded proteins for degradation. Consequences of misfolding. 1. Loss of function. Misfolded proteins may lose their biological activity. 2. Aggregation. Misfolded proteins can form aggregates, leading to cellular stress and disease. 3. Disease association. Protein misfolding is linked to various diseases, including Alzheimer's, Parkinson's, and Huntington's. Key concepts. Chaperones assist in protein folding and prevent aggregation. Proteostasis, the cellular network that maintains protein homeostasis, including folding, degradation, and quality control. Conclusion. Key takeaways. Protein synthesis involves transcription and translation. mRNA processing is crucial for mRNA stability. tRNA delivers amino acids during translation. Regulation ensures precise protein production. Protein folding and quality control are essential. Understanding the genetic control of protein synthesis is fundamental to comprehending cellular function and genetic diseases. These complex processes are tightly regulated at multiple levels to ensure that proteins are produced at the right time, in the right amount, and in the right location. Further research in this field will continue to provide new insights into the mechanisms of gene expression and their impact on human health.